All right, well, welcome everybody to, this is a data science primer designed for healthcare professionals, and I know in some cases budding healthcare professionals pursuing a career in data science. So my name is Dr. Patrick Dunn. My day job, I work for the American Heart Association in the Center for Health Technology and Innovation. We kind of are at the connecting point between the technology world and the clinical and scientific world. I'm a clinical exercise physiologist by background and training. I've worked in the field of cardiac rehab for almost 40 years now. I'm also I'm a continuing faculty member at Walden University, and I teach research methods and statistics, and I mentor doctoral students working on a PhD in public health and a Doctor of Public Health degree. I'm also, more recently, I have been trained as a software engineer. So again, it's kind of multi areas of focus for me. This course is going to be broken into 4 sections. Tonight we're going to talk about research methods and really the basics. If you're already a savvy researcher, this will really be a review for you. But I think in many cases, a lot of the mystery of research and statistics is just not understanding some of the basics. So really this is intended to really help you understand the basics, learn some of the terminology. In 2 weeks from tonight, on July 16th, we're going to begin part 2, which will be on the statistics and statistical analyses. Part 3, which will be, I believe, 1 week after that, so on July 23rd, we'll be really doing using software in research and statistics. And I'm going to show you a couple of different programs, including some basics in Excel, which many of you probably already know and do pretty well. I'll show you some examples in a program called SPSS, but then really show you some of the basics of how to do research in a program called R and another program called Python. And both R and Python are open source, free programs. So unlike SPSS, where you have to have an account to access the program, these are programs that you can download and play with and explore on your own. The 4th section, which will intended to be 1 week after that, so July 30th, will be on academic writing. So it doesn't do you any good to do your studies, to do a good statistical analysis, use the appropriate software, and then not be able to get your paper published. So we'll talk a little bit about both grant writing and academic writing, which is frankly very different from how you might compose an email or write a letter to your friends or colleagues. So how do you write in a way that can get it published in a journal? So I'd like to start with some introductions, and I know many of you are in the room. I think we do have a couple of people online. So why don't we start with the people online, and what I'll do is I'll just call your name out, and no need to get nervous. Really just your name. Maybe because some of you are calling in, name and where you're located, your interest in data science, some of your activities, your plans for the summer, and I want you to especially highlight fun summer activities, not just learning stats, but what are you going to do this summer for fun? So I'm just going to go down the list from the list I have. So I see Bilal Zia. Are you online? I can't hear you very well. Is anybody else hearing him?
No, I'm not either, but I can do Bilal's introduction.
Okay.
Bilal is an MSL, so he works in big pharma, prior to that, he was a researcher, actually at Houston Cardiology Consultants in my organization. And then he worked at Methodist in GI and liver diseases. And then he moved on to Lilac Pharmaceutical as an MSL in dealing with NOACs and factor new medications and factor 11 inhibition for a NOAC.
Great. Thank you. And below, if you can just put in the chat area, if you can, again, just some of the other things, what your plans are this summer, and some fun things you're going to do this summer. And for the rest of you, feel free to use the chat. I know for those of you in the room, it might be a little bit hard to do that. Next one I see on my list is Jonathan Reyes.
Yes, I'm here, Dr. Pat.
Yep.
Can you hear me?
Yes.
All right. Okay, well, one of my interests in data science is to perform not only objective studies, but also clinical trials. And for able to do that, I know that I need to master the data analysis. Like, as you said, Python program, RStudio, or SPSS are very important. So my data collection analysis is one of my interests. And also for summer activities, because I'm a medical student, I'm basically rotating. That's part of my summer activities, studying, preparing for my STEPS exams. And for summer, for fun, I usually love doing, I love running. It's one of my preferred activities, like feeling, I know it's hard to do that outside here in Houston, but it's like more one of my favorite activities for sure.
Thank you.
Thank you.
Next is Nadia. Nadia, I think you're on mute if you're trying to speak.
Hi, sorry, I was on mute.
Yep.
I'm Nadia Lee. I'm a high school student at St. John's, and I'm going to be a senior in the fall. And this summer, I've been doing an internship at Methodist Hospital, where I've been shadowing Dr. Chevre, and he's a plastic surgeon.
And what are you going to do for fun?
For fun, I've been playing lacrosse, I've been going on runs, and I also play guitar.
Excellent. Next is Rowan.
Hello, my name is Rohan. I am going to be a third-year undergrad student at the University of California, Santa Barbara. I'm going to be working with Dr. Dunn this summer as an internship, looking at how data science can inform personalized treatments in healthcare, specifically as it relates to cardiovascular disease. And for fun summer activities, I'm going to Colorado with family.
Excellent. And Umair? Oh, not able to speak? Okay, we see your response here. Okay, excellent. And now we'll turn it over to Dr. Ali. I know everybody knows you, but if you can start it off and then go around the room and have everybody introduce themselves.
Sure. So I'm Asif Ali. I'm a clinical assistant professor at University of Texas Health Science Center in Houston, Texas. I just accepted another adjunct appointment with University of Houston. So I'll be starting that next month. I am conducting a new organization called SINA Research Institute. So you'll meet some of my medical students and interns. We have about 4 medical students and about 6 interns at any time. And we are having a great time churning out papers and poster presentations and really clinical research. And we're really bridging this gap where medtech companies who need clinical research validation, we've created a company that helps bridge that gap. And I'm going to send my computer around the room so you can meet everyone else.
Hi, my name is Sami Faruqi. I am a rising senior at the University of Texas at Austin. I'm also a lead research coordinator here at Senior Research Institute. My interest in data science, as we're churning out research and papers, we find ourselves doing statistical analysis tools, and we're looking online how to do them. I think data science is becoming an increasing part of my life, and I want to get better at it if I'm going to continue. Summer activities. I love sports. I love to box. I love to play basketball, go on a run from time to time. I know I agree with Jonathan. It's a little too hot out, but I don't know what that is. But yeah, I'm looking forward to this. I'm going to pass it over.
Doctor. My name is Abdullah Saman. I'm a Jordanian doctor, graduated from Jordan and applying to the residency for internal medicine this year in September. I'm interested in data science because it's really important to be a doctor and also a researcher at the same time. I love to play tennis and many summer camp, refugees camp volunteering.
Hello, my name's Cheyenne, and I'm going to be a rising junior at St. John's this fall. And my interest in data science comes in, I'm trying to get more experience in doing clinical research, and I feel that data science is vital for understanding that and moving forward in research. And this summer, I've had some fun traveling, and I recently just came back from South America.
Hi, my name is Dean Palavan. I'm a rising freshman at Houston Christian University, and my interest in data science is I really would just like to know and be able to help out on more of the research that CRI is doing and conducting in office. So just in some summer activities I do boxing. I also like running in Memorial Park we have out here, and then yeah, that's pretty much it.
Hi, my name is Imad Khalid. I'm a rising junior studying public health at Texas A&M University. So this summer I'm a clinical researcher here at CRI, and some fun activities. I like playing basketball, and this summer I'm going to be hosting a basketball tournament for a charity that I'm really involved in. So I'm definitely looking forward to that.
That's it, Dr. Dunn, for those in person in this room. We still have. US is going to take a second.
Did we get everybody?
One had to unfortunately step out, but everyone else in this room is covered, sir.
Okay. All right, give me one more minute here. I'm putting in the chat, which won't be as helpful to the folks in the room, but I'll get it to you via email, are some files and some documents that'll be helpful. So give me 1 second. I'm going to upload one more document. And then we will begin. Okay. So I just put for resources, and I'll speak to them more towards the later, but I put in a Word document and 2 Excel documents into the chat, and I'll send them out as an email afterwards. The Word document really are my notes, so don't feel like you have to take a lot of notes here. Just listen and absorb. And if there's a concept that you're needing to learn more about, then you can refer to the Word document that's in that attachment. That being said, feel free to stop me if you have questions. Also, feel free to use the chat to speak among yourselves or to ask questions. I have a lot of material that I'm going to go through, so I'll be pretty focused on doing the presentation. So if you do have a question that you do need me to stop and answer, don't hesitate to interrupt me and just say, hey, Dr. Dunn, can you please say more about this or explain this or whatever we're talking about. Okay, so with that being said, it was a pleasure to meet all of you. It's very nice. We have a very diverse group. From doctors to high school students. So I will try to, get the tone of this content right, so that I'm not going too fast or too deep for some of you, but also being too basic for others. But do keep in mind the intention here is to really help you understand and learn the basics. It's, you can get a doctorate in biostatistics. So it's not intended to turn you into a doctoral level biostatistician in 90 minutes. That would be unrealistic. So the goals for tonight really are to learn some of the basic concepts used in health research, understand study designs and variable types, learn how to design a study, and then I'm going to show you how to download some of the software. It's not required for this course. You can always just watch and use the video. So depending on the setting that you're in and your comfort level with downloading additional software, feel free to do so. And there's several. I'll show you how to download R, Python, and a couple of different forms of SQL. And I did put the sample data set in there. We're going to be using the sample data set throughout the course. It's called Cardio, and we're going to be using it in Excel, SPSS, R, and SQL and Python. All right. So first of all, and again, what I'd like you to do here is just put your responses in the chat. But just some basic pre-assessment questions. What is your level of understanding with the basics of research? The next question, have you ever participated in a research study? Not as a participant, but as an investigator. And then finally, are you familiar with software tools for research? So I'm going to go ahead and move on here. So when we talk about data science and research, there's a lot of different areas, a lot of different levels that we can be talking about. So we talk about data science, we talk about research, there's public health research, we also have things like epidemiology, informatics, and both statistics and biostatistics. So I want to kind of walk you through what those are and what they mean. And first, I want to take a step back and really talk about evaluation methods to begin with. And there's really three forms of evaluation methods. There are qualitative measures, quantitative, and mixed methods. And while we're talking about data science here, I think you become a better data scientist when you understand even the role and the purpose of qualitative methods. So we're going to start with that. So a qualitative study is really used for inductive reasoning. And it's when the information is incomplete and you're really looking for deep insights into the lived experiences, attitudes, and behaviors. There's really four main types of qualitative. There's phenomenology, grounded theory, ethnography, and case studies. This is the design that I actually used in my own doctoral dissertation. I actually did a qualitative study on the development of health literacy skills. This was a grounded theory study, and really some of the basics are theoretical sampling. So a qualitative study doesn't sample in the same way that a quantitative study does. It's a smaller sample size, and you're almost hand-picking your sample, looking for those that can give you the best insights. It's just a different way to look at your data. Constant comparison is really how you code, and it's really is the insight that you're gaining here, is this something new, Or is this something that's part of another category that you've already explored? The coding can go from coding to categories and eventually to themes. And then eventually what you're looking for is what's called saturation. So in quantitative research, you're looking for power. You're looking for statistical power. In qualitative research, you're looking for saturation, which is you're really not learning anything new. You're kind of hearing the same thing over and over again. So an example of a qualitative study exploring the experiences and perceptions of using a telemedicine platform for chronic disease management. So a group of participants with various chronic diseases who are being asked to use a telemedicine platform. You conduct in-depth interviews and individual interviews with participants to gather their insights. So your data analysis, you're going to transcribe the interviews and focus group discussions. Use thematic analysis to identify the common themes, patterns, and insights, and then code the data into categories to analyze how they interrelate. Your findings might be, how these themes emerge and how people use telemedicine. And really, you might learn how they are satisfied or dissatisfied with the experience. And then from that, you can form conclusions, which may help you form better programs. It may help you form better hypotheses for your quantitative research. Now moving into quantitative, quantitative is deductive, and it's done when more is known about the topic. With quantitative, you're going to test a hypothesis, which may be establishing relationships or testing differences. You'll do descriptive statistics, including measures of central tendency and measures of dispersion. You'll use graphical representations, things like histograms and bar charts, and you're going to use, which we're going to explore next time, the concept of inferential statistics. Some of you are in epidemiology. We all learned about epidemiology during the pandemic. Epidemiology is the basic science of public health. Focuses on population health versus individual health. So you emphasize prevention and control versus diagnosis and treatment. The emphasis is on determinants, things like risk factors and even social determinants. A lot of work is done in surveillance and screening. A good example of that, we learned with the pandemic are what are called outbreaks and epidemics. So an outbreak refers to the occurrence of cases in a particular population that's greater than normal. So for example, COVID started with an outbreak in Wuhan, China. So it started locally, and they started noticing something new and something different. But even some other, SARS and Zika, all of those start as an outbreak where all of a sudden it's new. Even the flu, influenza, can become an epidemic. You might have like the swine flu or H1N1. And so it can go from an endemic to an epidemic. So an epidemic refers to the occurrence of a particular disease in a population that's significantly and statistically significantly greater than what's normally expected in a group or a population. The term endemic refers to the presence of a disease that's kind of always there. So influenza would be an example of that. Heart disease would be another example of that. So you have what's called an endemic rate, and when you exceed that endemic rate, then it moves into what's called an epidemic. And if it becomes global, if it becomes worldwide, then it's called a pandemic. You also, many of you clinicians, you may hear the term informatics. So informatics is looking at things like electronic health records, health information exchanges, conducting data analysis on telemedicine, clinical decision support, public health, bioinformatics, and patient engagement tools. And then finally, what is biostatistics? Biostatistics is a branch of statistics that applies to statistical methods of biological health and medical sciences. It's the analysis and design and interpretation of studies. What you're doing is you're going from a population to a sample, and you're using that sample. So for example, a population of blood pressure readings would be every blood pressure in the world. A sample would be a small subset that you would do in a study. So when you're doing statistics, you're working with samples. When you're working with populations, you're working with parameters. You have, again, with population refers to the entire set of individuals, items, or data points of interest. A sample refers to a subset of the population that you can use for observation and analysis. And the key concept is inference. So you're using what's called inferential statistics. You're inferring from that sample what you would see in the larger population. So let's talk about some of the basic concepts that are used in inferential statistics. The first one is probability. So it's a fundamental concept in statistics, a p-value. The p refers to probability. So it's the number of favorable outcomes divided by the total number of positive outcomes. So if you're flipping a coin, what is the probability of getting a heads? So feel free to shout it out or put it in the chat. What is that probability?
1/2.
1 and 2, that's right, because there's only one heads and there's two options. So it's 50%. What if you flipped it a second time? What is the probability of getting heads twice? It would be 1/2 times 1/2. So 1 times 1 is 1, 2 times 2 is 4. So it would be 1 in 4, or 25%. All right, so what if you're playing cards? What is the probability of being dealt an ace in a deck of cards? So how many aces are there? There's 4. There's 4 aces in a total of 52. Now what is the chance of being dealt an ace of hearts? How many ace of hearts are there? There's only 1 ace of hearts, so it would be 1 in 52. Now this will be a trick question. What is the probability of getting 4 aces in a row? So if you're dealt the first card, what is the chance of getting an ace? So it's 1 in 52. What is the chance of getting that second card being dealt? So how many aces are left?
Well, there's one less card as well.
There's 1 less card, so there's only 3 aces and there's only 51 cards. So it's 1/52 times, or 4/52 times 3/51. What is the chance of being dealt the third ace?
2 over.
How many aces are left?
2/50.
So 2/50, and then the fourth one would be 1/49. You multiply those 4 fractions, and that gives you the probability of being dealt 4 consecutive aces. So that's how probability works. Another key concept is a normal distribution. So when I lived in Dallas, I had about a 75-minute average commute time. So my mean was 75, and the standard deviation was 15 minutes. Okay, so I've broken this chart down by standard deviations. In a normal distribution, about 68% of the range is going to be plus or minus 1 standard deviation. 95% of the scores are going to be 2 standard deviations around that mean. So if I have, if my commute time today was 70 minutes, that's well within that confidence interval. That would be a normal time. If I had, maybe there was an accident and it took me 110 minutes, that's outside of that range. So that's how important it is to understand a normal distribution. There can be other distributions. So on the left side on the lower right, you could have a skewed distribution. So this one, skewed. The higher numbers are to the left, the lower numbers are to the right. Above that, you have a multimodal distribution. So you have literally within this, you're actually seeing 3 different normal distributions superimposed upon one another. That's why it's so important to know that you have a normal distribution. Another concept is linearity. So this is the relationship between two or more variables. It refers to the relationship between two variables where a change in one variable is proportionate to the change in another variable. So in the top graph, you see a linear relationship. So you can predict y if you know x. The equation here is y equals 3x. And your precision on that is how close those data points are to the line. In the middle graph, you have an exponential relationship, so you have to use some additional math to get to be predictive. And on the bottom, you have a concept called heteroscedasticity. So what you're looking at there is you see how the variance on the left side of the graph at the lower level, there's very little variance, but as you go to the right, the variance increases. So homoviscosity would be what you would see in the top graph, where the variance is constant throughout the range, and heteroscedasticity is what you see on the bottom, where that variance is changing throughout the range. Another important concept is variance. So it's the statistical measure that quantifies the degree of spread or dispersion around the data point. So how much of a variation do you have from one data point to another? So you use descriptive statistics. You have measures of central tendency, which are the mean, the median, and the mode. The mean is the average. The median is the middle point, and the mode is the most common point. The measures of dispersion are the standard deviation, the variance, and the range. Another important concept is the standard error of the mean. So the definition of that is the standard deviation over the square root of n. So if Dr. Ali is doing a study with blood pressure, and he has, say 10 patients versus another study where he has 1,000 patients, it's going to take the n or the number into count there. He's going to be much more precise with that larger n than he would with the smaller n. Another important concept is sampling. So you can have what's called convenience sampling, just the first come, first serve, whoever you can get to be in your study. My field is cardiac rehab, and if I can just get 10 people from my program to sign up, that's a convenience sample. A random sample is you randomly choose who's going to be in the study and who's not, and randomization is really important for eliminating bias in a sample. If you're relying on a convenience sample, maybe certain groups are being excluded for certain reasons. So random sampling is preferred if possible. And a third example is stratified sampling. So maybe you need to have a proportionate number of males and females, or proportionate number of age groups, or race and ethnicities. So stratification, and these are not always mutually exclusive. You can have a randomized stratified sample. It can be a convenience sample that's stratified. So it's just a way of ensuring that you have the numbers in the right categories. Now let's move to the variables. So you have roles of variables first. So you have a variable in studies. Typically, you have a dependent variable. This is the disease or health outcome of interest. And you have an independent variable, which is the predictor or exposure variable. So if we're doing a study on sodium intake and blood pressure would be the dependent variable, and sodium intake would be the independent variable. You can also have control variables. You may want to be controlling for age, gender, race, or type of condition. You can also have mediators, which is a third variable that may affect that relationship. And a mediator needs to be within the causal chain. It can't just be some random variable. You can have confounders, which are variables that affect the relationship between the variables. So for example, obesity confounds the relationship between smoking and heart disease, because there's a lot of very thin smokers that develop heart disease. So it confounds that relationship. If you control for smoking, then obesity becomes a much more robust predictor of heart disease. And a third kind here are moderators. Moderators are a third variable that affect either the strength or the direction of the dependent or independent variable. You also want to look at the type or the level of measurement. So variables are typically in 4 types. They can be nominal, which is a categorical variable. So nominal means a named variable. So gender, political affiliation, the school that you attend, those things are all nominal variables. Ordinal variables are rank order. So if you're using a Likert scale, maybe going from 1 to 5, but also your grade in school, freshman, sophomore, junior, senior, those can be used as ordinal variables. The last two are continuous or scale variables. So the first one is called interval. Interval includes variables that do not have a true zero. So temperature and weather can be an example. The number can be below 0 or above 0. So the 0 is just another data point. Ratio variables are the opposite. So 0 is a true value. So your reading level, your age, your weight, you can't have a negative age or a negative weight. So it starts with 0 and moves up. Another important thing about variables are variables versus constructs. So a variable is what we just talked about. So data points, so your blood pressure, your gender, your age. And you don't need to use a reliability instrument. You have to have good data collection techniques. But constructs are a little bit different. They do require reliability analysis. So if you're using an assessment to determine things like depression or self-esteem or trust or literacy, then you need to validate those measures. Okay, so it's always important to understand the difference between a variable and a construct. Okay, so let's pause here. And feel free to use the chat. Feel free to just shout it out. So what are some key concepts that we just discussed in research and statistics? Anybody?
The variables?
Yep, so normal distribution, the types of designs were, types of variables, yep, different types of measurements. So yeah, so think about, and again, always look at, things like normality. Do I have a normal distribution? Linearity. Do I have a linear relationship? What is the probability? So those are key fundamental concepts. And then what kind of variables am I looking at? Am I looking at a categorical variable, an ordinal variable? Is this variable a dependent variable, an independent variable? Is it a control variable? So those things. Yes, the N, and we're going to get into sample size and power in this next section. So to determine that, again, we're going to go back to some real basics here now, but these are fundamental and super important. The whole purpose of doing a research study is to answer a research question. So the most important thing you can do in a research study is come up with a good research question. If you have a bad research question, you're going to have a bad study. So a research question needs to be clear, focused, and concise, and it really guides the direction of the research. It defines the scope and direction of the study, and it helps you determine how you're going to investigate. So a good research question should be specific, researchable, significant, and clear and unambiguous. Related to the research question is the hypothesis. So a hypothesis is a testable statement or prediction about the relationship between two or more variables, and it's formulated based on the existing knowledge, theories, and observations. So a hypothesis also needs to be specific, measurable, and falsifiable. In other words, testable. And there's two types of hypotheses. There's the null hypothesis, which states that there's no relationship between the variables being studied, and it serves as the default position or statement. It's what you're testing against. The opposite, literally the alternative, is the alternative hypothesis, which suggests that there is a relationship between the variables. So again, you're always testing the null hypothesis, and sometimes that can be a head scratcher. Why am I testing the lack of the relationship? Well, think about it. It's hot outside today, right? Everybody would agree with that. But you think that it's hot today because you have a default, you know what not hot feels like, okay? And so today, if it's 96 degrees, you might your null hypothesis is anything greater than 80 degrees to me is hot. It's all relative because you might live in different parts of the world where maybe this doesn't seem so hot. Same thing. I have a new puppy named Toby. I think Toby's pretty cute. But again, if I don't have a null position, if I don't know what not cute looks like, I can't really say that Toby's cute. Does that make sense? It's super important to understand that relationship and understand the concept of a null hypothesis.
So the null hypothesis would be Toby's cute. The alternative would be Toby's not cute.
No, it's the opposite.
Oh,
the null hypothesis is Toby's not cute. Toby is a very plain looking dog. And the alternative is Toby's cute. Or we can say with the weather, we can say today is not hot. That would be the null hypothesis. The alternative hypothesis is it's pretty darn hot today. And so what we would do statistically is we would test the temperature. We would use that normal distribution to determine whether today is statistically significantly hotter than our null position. Or we would have a construct. Again, that's a good example of a variable versus a construct. If I had a construct that told me that Toby was cute, I would have to then test it against the not cute argument. So that's the whole concept in any research study. When they say the study was statistically significant, yada, 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 at a p-value, what they have tested is the null hypothesis. And they have they have rejected the null hypothesis. So I'm going to reject the null hypothesis that today is not hot. Therefore, I can accept the alternative that it is hot. And think about it, you really can't do the reverse. You can't test the position. You can form it in your mind that it's hot, but you can't really test hot without knowing the not hot. So that's why the null hypothesis is critical to being able to do the study. So if you're doing a study on a drug to lower blood pressure, again, you're testing that against the null that this drug has no effect on blood pressure. And then you test it, and then you may, if the drug is effective, you'll reject that null hypothesis and accept the alternative. but you can't do the reverse. You can't test the alternative, and so anyway, that's the importance of the null. So here's an example. We have a research question. Does a new vaccination program reduce the incidence of influenza in the community? So the null hypothesis is the new vaccination program does not reduce the incidence of influenza in the community. That means any observed difference in influenza is strictly due to chance. Okay, it had nothing to do with the vaccination. It's just simply the chance of these numbers changing. The alternative hypothesis is that the vaccination program does reduce the incidence of influenza. This suggests that the program is effective in lowering influenza rates in the community. So that would be an example of how a research question and a hypothesis are used in a research study. Now, because we're dealing with, we're not dealing with certainty, we're dealing with probability, there's always a chance that we're wrong, okay? So that's where what's called a type 1 and a type 2 error come into play. So a type 1 error occurs when the null hypothesis is true but is incorrectly rejected. So if a medical test indicates that the patient has a disease when they actually do not, that's a type 1 error. So if you do a stress test on a patient and it's positive, it means they flunked the test. You take that patient to the cath lab and there's no disease, that's actually a type 1 error. The type 2 error is the opposite. It occurs when the null hypothesis is false, but it incorrectly accepted, so it didn't reject. So in other words, the test fails to detect a disease in a patient who actually has it. So the patient who passes their stress test with flying colors and then drops dead of a heart attack on their way out of the office, that would be an example of a type 2 error. These two types are inversely related and they require balance. So what do I mean by that? So that's where power in quantitative research comes in. So you need power, refers to the probability of finding a statistically significant result. The minimum accepted value in typical research is 0.8 or 80%, which means that we would expect 8 out of 10 times to have the same finding. So again, remember, this is not 10 out of 10 times, it's 8 out of 10 times. And there is a relationship between, if we increase the power, we are making some trade-offs here. It does need to be, so power is calculated a priori. A priori means before we do the study. So it's estimated. It's estimated based on the literature. It may be done with a pilot study, but it's used to determine the appropriate sample size. If the sample size is too small, you may have the incorrect power and it may cause you to not have a statistical finding. So when you hear the term, the study was not powered to answer that question, that's code for the sample size wasn't large enough. So the power analysis, again, it's a priori. It can be done post hoc in the discussion to help explain certain things, but the key components are the alpha, the p-value, and the effect size. So we're going to talk about that next. So the alpha is the risk of a type 1 error. So the alpha and the risk of a type 1 error are the same thing. The alpha is the level we accept as researchers to decide to accept before we will be confident enough to release the finding. It's predetermined. It's typically 0.5 or 5%, and it's not calculated. It's something we choose. The beta is the risk of a type 2 error. The acceptable level for a type 2 error is 0.2. The mathematical relationship between power and beta is power is 1 minus the beta. So a beta of 20% means a power of 80%. The p-value, again, p refers to probability. is the measure of how confident we can be that the observation is true. So the p-value is calculated and it's compared to the alpha. So if the p-value is less than the alpha, you will reject the null hypothesis. For example, the alpha is 0.05 and your p-value is 0.03, you reject the null and accept the alternative. If your p-value is greater than your alpha, even if it's 0.051, then you can't reject the null. You might have to repeat the study, but you can't change the alpha once you've done it. That's literally cheating. Another important concept is effect size. It shows the size or the magnitude of the outcome that you're expecting. And there are statistical tests used to determine effect size. The most common is called the Cohen's d test. There's also the eta square, and a lot of you in a regression may be familiar with the R squared or the R2. So those are measures of effect size. So for example, if you're doing a home blood pressure monitoring program and you expect a 5-point reduction in systolic blood pressure, that's your effect size. Your effect size is 5. You may also be using confidence intervals instead of p-values. You're looking at those distribution points. So let's look at an example. This is my example for my commute time. So my commute time is 75 plus or minus 15 minutes. Okay, so I came in at 102 minutes. I would not reject my null hypothesis here. All right, now I'm going to compare during rush hour. And so on the left is my non-rush hour time. On the right is my rush hour time. And you can see there's very little overlap between those two curves. So in this case, I would be rejecting the null hypothesis. But if I compare my morning commute to my evening commute is greater, but there's a lot of overlap. You can see a lot of overlap in that mid-range. So if you have a point right here, which distribution does it belong to? You can't tell. Therefore, you would be not rejecting, or you would not reject you would fail to reject the null hypothesis. You also, another tool in your tool chest are using frequency tables to conduct ratios or tests. So things like the odds ratio, the relative risk, the sensitivity, and the predictive value. Okay, so I put in the chat the sample data set. We'll be using this throughout the rest of the course, and we'll use this as some practicing later on. One thing you'll do with calculations are to do rates. So you may have like an incidence rate. The incidence is the number of new cases compared to the population. So if there's 2 heart attacks per 100,000 people this year, that's the incidence rate. The prevalence rate is the total number of existing cases. So the number of people with heart disease in the population, that's going to be a much bigger number than the incidence rate, because there's going to be people who didn't have a heart attack this year. So that's the difference between incidence and prevalence. And then also you can have mortality rates. So again, but what you always want to be looking at is the rate. Is it rate per 100,000, per 10,000, per 100? That's super important, but they typically put it at a level where you have at least one number to the left of the decimal point. Okay, so you may do some calculations. So the first calculation to look at is the odds ratio. The odds ratio is looking at the exposure versus the outcomes. So a classic case of the odds ratio is the relationship between smoking and lung cancer. So you have smokers and non-smokers, and you have positive for lung cancer and negative for lung cancer. So and I'm going to I put the spreadsheet in my in the chat that says odds is I'm going to pull it up here. It's a way for you not to have to memorize these equations, okay? So this is the odds ratio. We're going to do 80, 20 for cases and controls, and 20 and 180. And we have, and so in this case, the odds ratio here is 36. So I've given you all a gift. I've given you the calculation. So save this spreadsheet on your computer. You never have to sweat through again doing the math to come up with that number. So that's the odds ratio. The next one is the relative risk. The relative risk is similar. Oops, hold on a second. All right, so we're going to do the same thing. So the relative risk was used in the Framingham heart study. So they're used in cohort studies. So we're looking here at exercisers versus non-exercisers, and we're using heart disease, positive for heart disease and negative for heart disease. So I can use the same spreadsheet here. I just have different numbers. I can put in 30, 170, 60, and 140. And what you have is, so the risk in the exercisers is 0.15. The risk in the non-exercisers is 0.3, and then the relative risk is the dividing these two numbers. So the relative risk in this case is 0.5. Okay, so we have a calculation for the odds ratio and for relative risk. So what's the difference and when do you use which one? The odds ratio is used in case control studies. Relative risk is used in cohort studies. We're going to go over those types of studies next. In the odds ratio, the focus is on the exposure of interest. So the exposure here would be smoking. In the relative risk, the focus is on the disease occurrence. So in the relative risk in the cohort, it's about who developed heart disease. The case control studies typically are retrospective in nature. The cohort studies are usually prospective in nature. The odds ratio is used best when the disease is rare and the exposure is more common. So lung cancer and smoking, whereas relative risk is where the exposure of interest is rare and the disease is more common. And then we have the formulas here. So this is just a screenshot of the odds ratio calculator that I just showed you. So it has the numbers and it has the formulas here. A related concept is we also want to know about the sensitivity and specificity and the positive and negative predictive value of a certain thing. So the sensitivity is also known as the true positive rate. So how it measures the proportion of actual positives that are correctly identified. The specificity is the reverse. It's the ability to predict the negatives. And again, that's why there's always a trade-off between sensitivity and specificity. But what you can get are the true positives and true negatives from this. So again, I have some examples here. It's in the same spreadsheet. So we're looking at the sensitivity, and it's with this next group here. So I already have this built in. So the calculus. So we're looking at true positives and false positives, false negatives, and true negatives. And so in this scenario, we have a sensitivity factor of 80% and a specificity factor of 90%, which is actually pretty good. You always want to look when you're, especially a diagnostic test, what is the sensitivity and specificity? And then finally, we also want to look at the reliability. So there's a test, it's called the Cronbach's Alpha. You'll see this reported in a lot of literature, and it's really how reliable is the construct in determining that variable. And so there's the Cronbach's Alpha, it's kind of like a grading scale. So the higher the better, the closer to 1, the better the reliability score. Okay, let's take a break again. What are some of the key calculations that are used in research?
Odds ratio.
So the odds ratio. So there's really for the odds ratio, relative risk, sensitivity, and specificity. So those are key. So you'll see those a lot in research studies. All right. Everybody doing okay? We're on the home stretch now. So now we're going to talk about the fun stuff. We're going to talk about study designs. So we have descriptive study designs, which can identify new problems, monitor trends in the group. They can be cross-sectional or longitudinal. They can be observational, used to collect data on exposure and rates. They can also be experimental. So descriptive studies, again, they can be cross-sectional, just a point in time. They can be both qualitative and quantitative. And the objective is really to describe what is occurring or what is going on. So example, the use of digital tools and technology among people with chronic conditions. So literally, what is their rate of usage? What is their age, gender? Really describing the population of people that are using digital tools. That would be an example of a descriptive study. Observational studies are used to observe and collect data on exposure to disease to determine an allocation and an association between variables. So observational studies can be cross-sectional or longitudinal. Cross-sectional studies are descriptive. Longitudinal studies can be either case control studies or cohort studies. All right, so what is a case control study? A case control study is used when cases are compared to controls. For example, participants in a virtual cardiac rehab program versus participants in a center-based program. Exposure of information is collected retrospectively. Often, you can do it prospectively, but if you're going to do it prospectively, you might as well be doing a cohort study. So the statistical test is the odds ratio. So an example, again, of our smoking and lung cancer. The odds of developing lung cancer if you smoke. So the cases are people who've been diagnosed with lung cancer. Controls are individuals who were not diagnosed with lung cancer. And the variable of interest is their smoking history. Positive, they're a smoker or a non-smoker. And obviously you can have, former smokers, quit, never smoked. But we've already done the odds ratio calculation, and the odds ratio is 36. That means you're 36 times more likely to develop lung cancer if you smoke than if you don't smoke. What about a cohort study? So a cohort study is done when participants are followed over time to assess whether they develop the outcome of interest. So in heart disease, the Framingham Heart Study is the best example of a cohort study. So you recruit from a population. So this is a population health study. This is not a randomized controlled study. This is a population study. It's the classification of exposure. Participants are divided into groups. And then they're followed over time. And you're going to use the relative risk to determine the results here. Okay, so Framingham Heart Study began in 1948 in Framingham, Massachusetts, to explore factors that contribute to cardiovascular health. It resulted in the identification of risk factors. This is where the concept of risk factors began in response to the rising epidemic of heart disease. So looking at individual factors like exercise. So what this is showing is that exercisers have a 50% reduction in their risk of developing heart disease compared to non-exercisers. And of course, they did this with all different types of risk factors, and that's where cholesterol, blood pressure, and smoking were identified as the three primary independent risk factors. The randomized control trial, the RCT, it's really considered the gold standard for testing the efficacy of new treatments or interventions. Drug studies or use RCTs to determine the ability to minimize bias and establish causality. So the randomization piece of it is super important for minimizing the bias. So it's, and in a typical classic case with a drug study, it's randomized, it's controlled, and it's also blinded. In some cases, like using digital tools, it's very hard to blind. So in my example here, a randomized clinical controlled trial of virtual cardiac rehab, you really can't blind because the participants know they're in the program and the observers know they're in the program. So it's not feasible. But you can do the other parts. You can randomly assign participants to virtual versus center-based. Going back to the cohort study, nobody was being assigned a group. Nobody was being assigned to the non-exercising group. Or nobody was being assigned to smokers or non-smokers. So that's the fundamental difference with an RCT. And then you follow, 3, 6, 12 months, and you're tracking your outcome of interest. And then the final thing, you may see what are called survival analyses. So survival analysis is a branch of statistics that deals with the analysis of time to event data. So in an RCT, you may have a survival curve. It's typically used to measure the time until the occurrence of a specific event of interest. This methodology is commonly used in clinical research, epidemiology, and even in reliability engineering to study the duration until an event occurs. So again, sometimes the timing is very important. And sometimes these curves will cross, and that's very important in your analysis. Okay, so one more break point here. What are some examples of study designs?
Cohort.
So a cohort study, a case control study, an observational study, a randomized clinical trial. Those are all examples of study designs. I want to end this by talking about a new concept, which is really kind of changing some of the rules in research and research methods. It's around artificial intelligence and machine learning. So, and Dr. Ali and I have co-authored a paper. It's coming out 2 weeks from today. We'll send you all a copy when it comes out. It's entitled an AI primer. So artificial intelligence is currently in use. It wasn't just invented 2 years ago. In fact, it was actually first conceptualized back in the 1950s. And it's used in many daily activities. I just bought a new car yesterday. Also, if you're going to get to see my new car tomorrow. There's AI all over the place. So there's computer vision. It tells me when I'm changing lanes. I can talk to it. I can ask it for directions. I can make a phone call without my hands leaving the steering wheel. All of that's done because of artificial intelligence and machine learning and natural language processing. Newer forms like ChatGPT, for example, are using what are called large language models. These are using generative, pre-trained transformer technology. We'll talk about that more in section 3. That was developed by OpenAI, but again, OpenAI was not the first company or organization to think about artificial intelligence. And all of the big companies, what really makes the game changer in AI now is the data. So a company that has a large data set and a company that has computing power. So those are the two key ingredients to the AI formula, is data and computing power. So what are the types of AI? So there are large language models. So these are advanced AI that are using deep learning, that are used in things like ChatGPT and then many other forms now. They're also used in computer vision, in automobiles, in medical imaging. They can recognize images and patterns. They're also used in voice assistants using natural language processing and speech recognition, so things like Alexa, Siri, the Google Assistant, and they use recurrent neural networks to automate repetitive tasks. So some examples of AI technology in healthcare would be the use of wearables like smart watches and devices, even like a home blood pressure monitor, for example, but also virtual, augmented, and mixed reality, and then chatbots. And again, the chatbots with large language models are just getting better and better all the time. In cardiology, there's a lot of use cases, making treatment plans more personalized, using more predictive analytics, using wearables and devices, the analytical techniques in imaging, in drug discovery, in robotics and surgical procedures, simulation, processing notes, analyzing the literature, analyzing errors and events, converting words to text to words and vice versa. So all of these are examples of using AI. And some of the results will be things like digital therapeutics, which would be the corollary to a pharmaceutical therapeutic. So these are digital therapeutics. Also remote monitoring, again using wearables and connected devices, and precision medicine, making things much more highly personalized. And they really work using what are called neural networks. So going from data inputs like, plans and journeys and data through the system to output, to outcomes, but really looking at not how these things are isolated and separate and independent, but how these different layers from social to medical to lifestyle to personal to digital are interrelated, and how one level can impact other levels. Some opportunities, using these for decision making, for improving diagnostics, for reducing the administrative burden, for developing new types of programming, for therapeutics and coaching, all cool, but there's also some concerns, the potential for bias, the potential for hallucinating, which is making things up, and the lack of transparency. All these can lead to being a vector of misinformation. Keep in mind that humans have these traits with or without AI. We all have our biases. We all intentionally and unintentionally make things up at times. And sometimes we're less than transparent with our motives and our actions. So these are just human traits that need to be monitored and managed. Okay, So the last section, just in the last few minutes, is I'm going to show you some examples of some software programs that you could consider downloading. Again, it's not required. You can just simply watch and observe, but you can explore some of these on your own. So the first one is a program, and I gotta get my notes out now so I'm more efficient with this. Give me a moment here. Whoops. I had to close my documents to share them, and so I have to open them back up. So give me a moment here to get reoriented. Okay. All right, I got my stuff together now. So G-Power is a program that's used for conducting a power analysis. So I'm gonna go out of presentation mode here. Yeah, let me just do this from my notes page. And you have this same notes page. So if I go to, so it's actually a German company. Okay, so you go to the, so this is what you would be looking at for your website. It works on both Windows and a Mac, and it gives you the instructions right here on how to download. And again, there's Mac and PC versions here. Let me go back to my Word document. I had a couple of links. So I'm on a Windows machine, so I can click here on this, and it's gonna put this on, download this file onto my computer, and then I would click to open, and I can install G-Power from that link. So that's how you would install G-Power. What does it look like? This is G-Power. So if I'm going to be running a t-test, and I'm going to be using 2 independent groups, and I'm going to be a priori, my effect size is 0.5, my alpha 0.05, I'm going to change my beta, I've got a 1-to-1 allocation, I'm going to calculate, and now I've got my sample size calculation. So that's how G-Power is used. I'll show you more about that in subsequent weeks. I'm just showing you now how to download it. There's also R. So there's a couple things to think about in R. So you can download R, and you're going to get this, and if you pull up your data set, I can pull up my cardio, and I've got, and I can do, I didn't do that right. There we go. I can do a summary of all of my data here. So this is one way to use R. The way I would recommend R is to download the R Studio. And you can download R Studio from this link here. There we go. Click here. And what R Studio is going to look like is this. So it's you're going to have different window panes in here. What I am going to also put in the chat is this YouTube video on how to download R and RStudio. There's also a program called Jamovi. So you can click on Jamovi and download. Jamovi is basically a much more user-friendly way to use R. So I'll show you more about Jamovi. But if you like programs, you don't like to use it from what looks like a computer code, then you'll use, what you'll do is you'll open up your data set. And now you've got your data set, and now you can run your statistical tests in here. So that's Jamovi. Next one is MySQL. So there's MySQL, there's also the SQL Server from Microsoft. They're both open up. So MySQL is open source and free. So you can click here and install. And what you're gonna want to install is the MySQL Workbench. And I'll go through tutorials on how to use all of these. But what we're going to learn are select statements. So I'm using this data set, I'm selecting all variables from this cardio data set, and that's how MySQL works. You can also do a program called SQLite. So there's a link here. So there's a SQLite Studio. And again, this is just a little bit easier, a little bit more user-friendly way to do SQL, but we can still learn how to do select statements from SQLite. SQLite you'll actually download right on your computer. And so it's much more self-contained. And then the last one, two more, is Python. So you can download Python, and you can download it from the command line, or you can download programs that you can use to run Python. So one program that I use is a program called Anaconda, and I'll show you how Anaconda works. But my suggestion, the last one I'm going to show you to download is Visual Studio. If you click on Visual Studio, and you can do for Windows or Mac. Visual Studio is a great program because it'll use multiple platforms. So you can use Python, you can do R, you can do, we're even going to learn a little bit about HTML and CSS, you can JavaScript, all of the programs can be run in Visual Studio. So if you're looking for a program to just learn some basics of programming, I would recommend Visual Studio. And then again, in section 3, we'll go into more of the details of how to use these programs. Not trying to teach you these programs tonight, just showing you where to download them so that you may want to get a little bit of head start in becoming more familiar. But if these things scare you, then don't worry about it. We will do them together. Really in section 3, I just wanted to give you enough time to start wrapping your head around it. The next presentation in 2 weeks, we're going to be talking about the statistical methods. We talked about the research methods and the study designs. Next time, we're going to be talking about the stats. So doing things like t-tests and ANOVA and regression and chi-square. And then I'll be moving into how to use these things in these different software tools. But then in section 3, we'll actually run these tests using the cardio data set in all of these different programs.